Welcome to medicinal biochemistry class on nucleic acid metabolism. Today we will learn about genetic code. At the end of this class, the student shall be able to define codon, define Wobble position, explain degenerate nature of genetic code, and write the whole genetic code. Okay. So, introduction to this genetic code. Introduction to genetic code. So, genetic code is a set of rules used by living cells to translate information encoded within genetic material. Okay, so these are the set of rules. So, this is used by cell to translate the information that is present in the DNA or mRNA sequences of nucleotide triplets. Okay, so they are present in the form of a set of 3 nucleotides, what we call it as a nucleotide triplets. So this genetic code, so this is a language in fact, or a set of rules that is used to translate the information that is present in the DNA or mRNA into proteins. So it carries the information. The genetic code precisely carries the information about what amino acid has to be added at a particular point of protein synthesis, at that particular point of protein synthesis. Okay, so the sequence of amino acid that is present in a particular protein is decided by the sequence of genetic code that is received during translation. Okay, so each group of 3 nucleotides on the mRNA is called as codon. So that the 3 nucleotides that are present will carry the information of what amino acid has to be added. Okay, so there are 4 bases. Okay, so since there are 4 bases in mRNA, that is adenine, guanine, cytosine and uracil, AGCU. So there are 4 bases and each codon is a combination of 3 nucleotides. Okay, by permutation and combination there are around like a 4 cube. It gives 64 possible codon. Okay, so 64 possible codon of, that is each codon carrying 3 nucleotides. Okay, so these 64 codons code for 20 different amino acids that are present in proteins. Okay, so genetic code is degenerate. We call that as degenerate because more than one codon is used for most of amino acids. That is, more than one codon can code for one particular amino acid. That we will see in the next future slides when we discuss when we look into the whole picture of genetic code. Okay, so since this genetic code is degenerate, you can't exactly say like which codon from, like you take out a particular protein and you take out a particular amino acid and you're taking out that particular amino acid, and you can't predict like which particular codon has coded for that amino acid or from where this amino acid has been coded. That is, you can't predict exactly the base sequence of gene it came from. Okay, in most cases the third base of the codon, so usually the third base of the codon is called as Wobble base, so that can be altered without changing the amino acid. Okay, so here AUG, that is when you have a codon AUG, adenine, uracil and guanine, so that is the start codon. So here when this like during translation, when AUG arrives, the translation begins, that is the protein synthesis begins, the translation begins. Okay, so all proteins are initially translated with methionine in the first position. AUG codes for methionine and that is the start codon, and the first amino acid during the translation is methionine, and methionine is often removed after translation during post translation modification. Okay. So there are also internal methionines that is nothing to do with this first position of methionine in most proteins. Again, they are also coded by same AUG codon. So there are 3 stop codons, also known as nonsense codon. When nonsense codon is come across, so that is UAA, UAG, and UGA, so the translation will stop. Okay, proteins end, that is in a stop codon. The protein synthesis end in a stop codon which codes for no amino acid. So these 3 codons, UAA, UAG, and UGA, do not code for any amino acid during translation. Okay, so this is the overall picture. So now I'll tell you like how to write this. So here you need to create around 4 into 4, 16 boxes. Okay, in 16 boxes, in each box there are around exactly about, okay, so this is the genetic code, the whole genetic code, typical genetic code, and here, so I'll tell you how to write this. So you have to draw around 4 by 4 boxes, that is 16 boxes, and each box will carry 4 genetic codons. Okay, each box will carry 4 codons. So on the left side, so you need to write 4, that is the first letter representing in the, that is represented in the codon. So that is and each box, in each box you should represent 1 nucleotide. So here U denotes uracil, C denotes cytosine, A denotes adenine, and G denotes guanine. And on the top again you have to write the 4. In each column you have to write 4 different nucleotides. So here again U denotes uracil, C denotes cytosine, A denotes adenine, and G denotes guanine. And on the right side, so here in each row you have to write all 4 nucleotides, UC, AG, uracil, cytosine, adenine, guanine. So the sequence should be maintained in all these 3 on the left side, top, and on the right side columns. Okay. So UC, AG, UC, AG, UC, AG. So here how to write this genetic code. So the first letter in the first box, so is U. And the second letter is also U in the first box, and the third letter is also U. So this is the first codon UUC. So here in the first box only the third nucleotide will change, that is again cytosine. The second codon is UUC, third codon is UUA, fourth codon is UUG. Okay, so now coming to the second column, the second box. So here the first codon is U in all the 4 codons, and the second codon is C, and the third codon again it will change. Okay, U, C, A, G. Okay, so the remaining genetic codons are written in a similar fashion. So you're going to write 64 genetic codons, and if you follow this method, you need not remember this. You can write it easily. Okay, so the only thing you should remember is what codon is going to code the specific amino acids. Like UUU and UUC codes for phenylalanine. So look here. So there is so 2 codes, that is one codon containing all uracil nucleotides and other codon containing 2 uracil and 1 cytosine. So both can code for phenylalanine. UUA and UUG codes for leucine. And so these 4 codons again code for leucine. Okay. So 3 codons codes for isoleucine, these 3. And AUG is the start codon, and it codes for methionine always. Okay. So and ribosome running through this mRNA comes across AUG, so the translation process begins. That is okay. So GUU, GUC, GUA, GUG codes for valine, and these 3 codons code for serine. So these, sorry, 4 codons, these 4 codons code for proline, these for threonine, these for alanine, and these for tyrosine, these 2, and UAA and UAG are nonsense codons, that is, they are stop codons. Similarly, UGA is a stop codon. Okay, so similarly, so there are other codons that are going to code for specific amino acids. Okay, so in summary to this genetic code, so if you look at this genetic codon, so more than one codon can code for one single amino acid. Okay, so all 20 amino acids are coded by these 64 codons. Okay, so methionine AUG is the start codon. So, and UAA, UAG, UGA are called as nonsense codons, that is, they are stop codons. Okay, so these are the important features of genetic code. Okay, so more about, so the genetic code is almost universal, that is, same codons, like this genetic code, same codons will code for the amino acids what it is there for humans, for other animals as well. Okay, so it is used for both prokaryotes and eukaryotes, but there are certain variations, like there are certain exceptions. So mostly in mitochondria, which have very few genes. Like for example, CUA codes for leucine in the universal code, but in yeast mitochondria it codes for threonine. Okay, so similarly AGA codes for arginine in the universal code, but in human and Drosophila, that is the mitochondria of human and Drosophila, it is a stop codon. Okay, so there are few known variants in the code used in nuclei, but in mitochondria there are quite a good numbers. Okay, so I have referred Wikipedia and also I have referred many websites. Okay, happy learning. Thank you.